If you've seen living cells under a microscope, you've probably seen them swimming around like this. And of course we know cells are moving around the body all the time. White blood cells travel around to combat foreign microorganisms. Muscle cells contract to keep our bodies moving. My focus in this video is to show how researchers quantify these movements and what we learn from studying cell movement. Of course, for a cell to move, whether it be a sperm cell or a white blood cell, it has to be exerting some amount of force, just like a person walking has to exert a force to fight gravity or air resistance, which is why the technique I'm discussing today is referred to as traction force microscopy. There are several variations of traction force microscopy and several ideas and methods of how to best capture the movement of the cell and the forces exerted by a cell. But basically it all starts with placing a cell on a material, a linear elastic material, and watching what that cell does to the material. The earliest form of traction force microscopy started in the 80s. It involved forming a thin film on the surface of silicone oil and placing a cell on top of the film and observing with a technique such as confocal microscopy how the film moved and wrinkled under the cell. This was a pretty rudimentary method pioneered by Albert Harris, David Stopak, and Patricia Wild at UNC. But even early on, before more quantitative methods were developed, this method could tell you quite a bit about different cell behaviors and relative strengths, say showing the difference in cell forces between a muscle cell and a leukocyte. But there's a lot of difficulty in trying to discern the precise forces and displacements in this method simply from looking at a couple of wrinkles. After all, some of these wrinkles are so much larger than the cell that it's really hard to tell what's even going on. In the current literature, the most widely adopted method is to use a hydrogel, such as polyacrylamide, sometimes called a PA gel, as the substrate, and you embed little fluorescent latex beads in the gel, which will act as displacement reference points, and then you polymerize the PA gel to the particular stiffness desired for your experiment. Now you can imagine the cell creating a stress configuration in the gel, and then you remove the cell using trypsin to image the unstressed state of the gel. Fluorescent beads, when being displaced by a cell, retain their shape and their movements are also well defined and easier to track than, say, wrinkles in a film. They show up well using any fluorescence imaging methods, such as FRET. By tracking the bead movement, we can calculate strain and back out the stresses of the cell. Also, PAGL, from what I understand, is pretty cheap and easy to work with, and you can tweak the consistency and stiffness of your matrix, which is why it's widely used for this method. Another promising method in the current literature is placing cells on top of little microneedles or microposts, and then calculating the forces of the cell based on the simple mechanics of how they deflect the post, all observed using scanning electron microscopy. You can also adjust post height or shape to test for anisotropy effects, or adjust the stiffness, for example, so it's quite a flexible method as well. All the methods I've described so far are the 2D methods, and they are the primary methods, the most popular methods, involving a cell on some substrate on a glass slide under a microscope. 3D techniques are currently mostly similar to 2D methods, with an added layer of complexity. Some of these so-called 3D methods are simply the same 2D setup, using more computationally intensive image tracking of these microbeads in a certain depth of field, and reconstruction of these 2D features in a 3D generated space. One of the more complex 3D methods involves embedding cells inside a collagen gel structure, and as it moves around, again using these more complex time-intensive visualization methods to generate a 3D strain map. So what have researchers learned from using traction force microscopy? Well, we've gained a better understanding of how cells move and the way certain cells exert force, what directions they exert force in, how strong they are relative to other cell types. Research has also begun to show how force stimuli can influence cell growth and cell differentiation. A few studies examined how mesenchymal stem cells, or MSCs, may spread or ball up depending on the stiffness of the surface substrate they rest on, and that this shaping can actually determine if the cells become osteoblasts, bone cells, or adipocytes, fat cells. Other works now also seek to eventually connect these cellular mechanics to larger-scale tissue formation and manipulation.